First of all, there are two types of chromosome, which are autosome and sex chromosome. This is the representative image of a typical human karyotype. The chromosomes that pair from 1 to 22 are called autosomes, which they control characters other than sex characters or carry genes for somatic characters. To be precise, they have got no function to deal with the sex of the individual. However, the last pair is the chromosome involved in sex determination, which is known as sex chromosomes. So, if the two are identical, which both are X and X, then the individual is a female. In another case, when one chromosome is X and the other is Y, the individual is a male. Next, I would like to introduce the homologous chromosome. Diploid organisms have two copies of each chromosome except the sex chromosome. Both the copies are ordinarily identical in morphology, gene content, and gene order. Each pair of chromosome is made up of two homologue. Homologous chromosome is inherited from separate parents. One homologue comes from the mother and the other comes from the father. Now, we proceed to the structure of chromosome. The chromosome at the left side is not duplicated, while at the right side is the duplicated copy attached to itself. So, these are the duplicated sister chromatids which are attached to the same chromosome. This is known as the P arm and this is known as the Q arm. So, how do we recognize which is the P and which is the Q arm? It depends on size. The smaller arm is the P arm, while the larger arm is the Q arm. So, the left-hand side chromosome has one P and one Q arm, while the right-hand side chromosome has two P arm and two Q arm. Now, let's focus on this chromosome structure. The structure which the arms are attached is called centromere. It is the portion of DNA that acts as a link between the two sister chromatids. Other than that, its major function is the attachment of spindle fiber during the cell division process. Moreover, the two ends of the chromosome are known as telomeres. They play critical roles in chromosome replication and maintenance of chromosomal length. The telomeres are highly stable and telomeres of the different chromosome do not fuse. The telomeric region of chromosome is made up of repetitive sequence of T and G bases. Next, the constricted or the narrow region found at any point of the chromosome other than that of centromere or primary constriction is known as secondary constriction. The chromosomal region between the secondary constriction and the nearest telomere is called as satellite, and chromosomes that possess this region are called as satellite chromosome or set chromosome.
Move to the classification of chromosomes. The chromosomes are classified according to the centromere position, where for metacentric, the centromere is located exactly at the center of the chromosome, and thereby, two arms are roughly equal in length. The metacentric chromosomes will appear as V-shaped structures during anaphase stage in the cell division. And as for the submetacentric, the centromere is located near the center of the chromosome, but not at the center. Thus, these chromosomes will have two unequal arms, a small P arm and a large Q arm. Submetacentric chromosomes will appear as L-shaped structures during anaphase stage of cell division. For acrocentric, the centromere is positioned at one end of the chromosome in such a way that it produces a very short P arm and exceptionally long Q arm. Acrocentric chromosomes appear as G-shaped structures in the anaphase stage of cell division. And lastly, the telocentric chromosomes. The centromere is located at the proximal end of the chromosomes. Thus, telocentric chromosomes are long rod-like chromosomes and appear as eye-shaped structures during anaphase stage of the cell division. As for chromosome studies, we can already appreciate how confusing the terminologies are, right? Especially the words chromosome, chromatin, and chromatids. So, first things first, the chromatin. Chromatins is composed of DNA that is wrapped around the protein called histones and is packaged into thin stringy fibers. During interphase 1, chromatin is in its least condensed state and appears loosely distributed throughout the nucleus. And as the DNA content is huge, the only way to place it properly in a small tiny nucleus is by properly condense and coil it. And this condensation is only going to happen during cell division, where the further condensed form of chromatin is called chromosome. So basically, chromosome is chromatin, but in a condensed shape. We have understand about the chromosome and chromatin, but what is chromatid? Okay, so chromatid is one copy of chromosome where it is either of the two strands of a replicated chromosome. The chromatids connected by a centromere are called sister chromatids. Lastly, what is the role of synapses and crossing over? Synapses is the pairing of two homologous chromosomes that occur during meiosis to form tetrad. Sex chromosomes undergo synapses. However, the synaptonymal protein complex that holds the homologous chromosomes together is only present at one end of each sex chromosome. As for chromosomal crossover, it is the crossing over between the non-sister chromatids within the tetrad, where they exchange different segments of genetic material to form two recombinant chromosomes. The diagram below shows that the event of synapses and crossing over occur in a sequence where the crossing over will occur immediately after the synapses. The exchange part of chromatids, where there's exchange of some distal portion of DNA of chromosome, will be called recombinant chromosome, while the part that is not involved in the exchange or crossing over of chromatids will be called non-recombinant chromosome.